So, day 4, this is going to include everything front and back. You will notice I wrote a lot more stuff this time. I was up pretty late writing on this thing to help you guys out because I know our time will be crunched. So let's go. Let's do this thing. First off, let's start off with, let's do genetics first. We're going to work clockwise here. Okay, so genetics. We've got some terms that we really care about. Allele. An allele is a letter that describes a trait. So the letters that describe these traits, thanks. Who does? There you go, Killian. The letter that describes the traits, we usually wrote them as like big letters or small letters, so like this or like this, right? Which leads us to the different versions of these, which are called dominant and recessive alleles. Dominant alleles mean they appear even if there's only, oh gosh, one copy. So we're saying in this case, we can have a heterozygote individual, big letter, small letter. The big letter wins. So whatever the big trait is going to show up. Recessive only appears if 2 copies are present. So the only way you'll ever see a recessive trait show up in the population is if we have little letter, little letter. Does that make sense? Okay. So we've got dominant, we've got recessive, then there's also homozygous. Homozygous means you have 2 copies of the same allele. So this is big A, or it could even mean little a, little a. Notice how they're both the same size. That's what we mean by homozygous. Remember, homo means same. And then we have heterozygous. Heterozygous is going to be whenever you have 2 different alleles. The only cases of this are big letter, small letter. We never put small letter in front of big letter. We always put the larger letter first. That's a key thing. Don't forget that. Now, hybrid, this one's easy. It's just another fancy way of saying heterozygote. That's all it really is. So just try to keep in mind, big A, little a is a hybrid. It means it has both of the different versions. And hopefully, this leads us to finding some pretty cool traits later on, a 3rd phenotype. So, doing okay? Do you have any questions? Ayo. All right, so let's talk about complete dominance. You guys ready? Okay. Complete dominance. Like I said, I've tried to write a lot of things here for you. I just want to remind you really quick. This means dominant wins, right? So in a heterozygote, the big letter is the trait that you'll end up seeing. I've even given you a little chart here. A thing to keep in mind is this is the standard operating procedure for genetics. We always assume things are complete dominance unless we tell you otherwise. And that would be true of any question anybody would ever ask you in genetics. They're not just going to expect you to know what form of inheritance to use. They will tell you, unless if they don't, you assume this. So with that, let's go ahead and look at our example that we have here. We've got a cross, and we call this a monohybrid cross because only one trait is being traced at the time. And we have big A, and we're going to cross that with a heterozygote, a big A, little a. If we see that big A is red, and of course little a is, let's say, a white flower, then in this case, we're going to see big A, big A, big A, big A, big A, little a, big A, little a. When we look at this, we can see some percentages that are worked out for us. What percentage are heterozygotes? 50. You're right. It would be 50%, and that would be these bottom 2. What percentage are going to be homozygous dominant? 50%. Well, it kind of gives away, we've already used 100% of our samples, so there is 0% that are homozygous recessive. Nobody would see the white color show up in their population. But when it comes to red and white, what percentage are red? 100%. Exactly, 100%, whereas white is at 0%. So even though we have different letter combos, we can still create the same appearance, which is why it seems that some traits seem to hide and skip generations. I should also go ahead and mention to you those words genotype and phenotype are used here. Okay, genotype is the allele combo. That's what these two-letter combos are, big A, big A, big A, little a, little a, little a. But phenotype is the physical appearance. Okay? What they look like. That's the phenotype. Do they look dominant or recessive? Do they look red or do they look white? That's what a phenotype is. Doing okay? Do you have any questions there? Remember, all the other forms of inheritance are based off of this. We just make some tweaks, some small changes to those rules. Doing okay? Okay, let's go ahead and do incomplete dominance. Incomplete dominance basically works the same way, but now whenever we have a heterozygote, in this case we're going to use the letters big R and little r, we'll see a new color appear. So, of course, R is still red. Little r, in this case, will be white. But when we blend the two together, when we form a hybrid, we will see a third phenotype, pink, right? So, let's do that first example. That's really small. So we got big R, little r, little r. Every single one of these is going to come out big R, little r. I'm not even going to put it in each individual box because I know every single one's going to look exactly the same. Knowing this tells me that 100% of them are going to be pink, aren't they? Right? So that third phenotype appears, shows up, and is in a high frequency. But my example down here, big R, little r, crossed over with another one just like it, I definitely have to use every single box here, don't I? Now this actually brings up some interesting points, and it says what percentage of these are pink? What percentage are pink? 50%. And you'll notice it kind of moves at this angle right here. That means 25% are red and 25% are white. I should just go ahead and take the moment to say we have these things called genotypic frequencies, or sometimes a genotypic ratio, and this means it's now at a 1 to 2 to 1 ratio, which shows dominant, heterozygous, dominant. Does that make sense? Okay. We can also do that for phenotype, and phenotype is what they actually look like. Whenever we now have a third phenotype, it makes the exact same ratio. This actually helps you speed up your predictions because you know if two heterozygotes are crossed together, 1 to 2 to 1 is the number that results. Doing okay? Questions? Okay, let's move on. Codominance. Codominance is almost the same thing as incomplete dominance, but instead of having an incomplete look, an unfinished look, and having a little bit of red showing up, this time we're going to have two dominant traits at the same time. So our third phenotype is going to look totally different. So in this case, we have two dominant traits. R is going to be red. Capital W will be white. It's not a recessive trait anymore in this case. And then if we get an R and a W, it's going to have both red and white. Why did I add an E to the end of this? I'm not sure. But it's going to have both those colors at the same time. Splotchy, like a Dalmatian or like a cow, right? So when we look at this, our simple example, we got R's and W's, R's and W's. So we got big R, big R, big RW, big RW, WW. When we look here, we can make predictions and we can kind of look at this. It says what percentage are going to be both, have both colors. I still added an E. Don't know why I'm doing that. Anyway, that's 50%, right? Because we can see that 1 to 2 to 1 ratio shows up. 2 out of 4 is 50%. But the interesting one, and I'm bringing this up because I have a hunch that they'll bring this up on the EOC one year, and maybe one year I'll be right. But there's blood types. That's what this I, little I with the A's and B's is all about. So if you see a capital I with an A and a lowercase I, or if you see a capital I and a capital I with two big A's there, that's A-type blood. Okay, so that you can be a heterozygote or homozygote for this trait. You'll notice that a little I is actually the O trait. Okay, which means it doesn't have A or B, it's just a recessive. So there can be somebody that's little I, which means they have O-type blood. If somebody has both A's and B's, then we call that AB-type blood because it's codominant. And of course, there's the same thing for B-type bloods. So when we cross this together and we have a heterozygote mom and a heterozygote dad, we can see some pretty interesting stuff. And if we look here, it actually shows that the percentage that are AB type blood both is what? 25%. Only one of these boxes has AB type blood. The other one has B, the other one has A, and the last one has O. So there is a way that an A type and a B type parent can make kids of 4 different blood types, which is pretty crazy, considering, you know, it might think some paternity tests need to happen, but it's absolutely very much possible. How you guys doing? Any questions there? You sure? Okay. Last type of genetics we are going to focus on is going to be sex-linked. Sex-linked is basically, remember how the X chromosome and the Y chromosome look different from each other? The X chromosome is really big, whereas the Y chromosome is really tiny. So it can't hold information the same way the X chromosome can. So typically, traits can fit on females that can't necessarily fit on males, which is why some females aren't as affected by diseases the same way males are. A good example is color blindness, which is the example that we have there. So you'll see me, I've labeled all the situations there. You'll see females can become carriers. Carrier means a heterozygote. Males cannot be heterozygotes because they only have one allele that carries the trait. So knowing this, we're going to cross a woman who is what we call a carrier, meaning she has one recessive allele, but she has normal vision. She just carries the trait, and her kids could potentially receive that. And then the male over here, he's going to have normal vision. So we have 2 normal vision people, no color blindness, and we're going to cross them up. You'll see I'm laying down my chromosomes first, and you'll see there's a 50% chance it's either male or female. Then I lay in my detailed alleles. So in this case, the female will have normal vision. In this case, the female born will be a carrier. In this case, the male will have normal vision because the Y has nothing on it. It's too small. So whatever the X chromosome is in this case, well, there you go. It's going to be that. And then, of course, this male will be colorblind. So the question on your paper says, what percentage of males are colorblind? It would be, but it's 50. The reason why is because it says of just the males, so there's only 2 boxes that are males. What percentage are colorblind? Half of those males. So that's 50%. If we would have said, is it children, you would be right. It would be 25%, but we are being specific here. I'm just pointing this out because if they ask you a question like that on the EOC, you know, oh, I should really pay attention to that part right there just to be safe. Anyway, there you go. Any questions on that? Let's talk about GCAT. Are you ready? Are you ready for some GCAT back in your life? Okay, good. So let's talk about that DNA, or nucleic acids in general. So I've written down just about everything on your paper that needs to be seen. I'm just going to highlight a few things. First off, G, C, A, T. G and C always pair together. A and T always pair together. Those are the letters involved in DNA. Guanine, cytosine, adenine, thymine. With RNA, it's almost the exact same thing, but instead of the letter T, we use the letter U. This is just because RNA works this way. So uracil replaces thymine. I should also point out they pair up on these things called Chargaff rules, and that's basically because of the shape of these molecules. But anyway, you will see that DNA is that double helix shape. Let's try that one more time. Right? That double helix shape is really, really important because it locks and secures things and makes sure things can't bond to it. And of course, it protects it. And this stuff stays in the nucleus at all times. It never leaves. It always lives inside of your nucleus. With RNA, it's just single-stranded, and it's only got half of those little branches that are found there. This is super important because, once again, this means it can bond with other things. It's also shorter and temporary, so it can get sent off and do things in other parts of the body because DNA doesn't move, does it? It stays in place. I should also mention that RNA has a few different versions. There's rRNA, there's tRNA, and there's mRNA. rRNA is ribosomal, which means it literally makes up the ribosome. Ribosomes are made up of rRNA. tRNA is called transfer RNA, which basically transfers the amino acids to the ribosome and actually links them together. And then there's the mRNA, which is the messenger RNA. This transmits the information from the DNA all the way to the ribosome. So it's like all three pieces are coming together in the process of translation, and that's where it occurs. Yeah, okay, cool. Central dogma, I'll squeeze this on here. Okay, so DNA, if we make more copies of this, we call that replication, right? When we go from DNA to RNA, we call this transcription. And then mRNA to protein, we call this translation. This is the one that uses the codon chart, translation. Do you guys remember your codon charts? Yeah, it doesn't mean you have to memorize it. If they ever ask you a question on the EOC, they'll give you a copy right there in the question. It might be the square version or the circle version. Just remember the circle version, you work inside out, which I think most people find that one to be the easiest. If it's the square one, remember you use the left side, then the top, then the right side. But once you know the side and the top, you'll find your letter combos. But anyway, this is like the central idea behind how genetic information flows inside of the body. I should also just mention to you, I gave you an analogy of like baking cookies for my birthday, right? And I said we had this family recipe that's in a family cookbook that we keep at grandma's house and nobody can take it because we don't want to damage it. That's like the DNA. The recipe that when you go over there and you hand write the instructions for making that recipe, that's like making mRNA, and then taking that mRNA to your kitchen and actually making the cookies from it. That is, of course, like making protein. So we have transcription translation in terms of analogies there. How you doing?
Tired.
I know. That's why I have coffee today. It's mostly chocolate milk, though. Anyway, okay. Let's go. Let's keep going clockwise. This will be fun. Transcription. Transcription is basically where it occurs, by the way. This is going to occur in the nucleus. So DNA can't leave the nucleus, so we need to make mRNA from that. And we said there's like this cool editing process, so we turn DNA into RNA, and RNA gets edited out. We cut out the we cut out the introns, we leave behind the exons, and we make mRNA. But anyway, this basically works like this. DNA gets transferred to RNA. If we have a few letters here, which in this case I wrote A, T, G, G, C, and I'm just going to get lazy and finish there. What does A always pair up to? U. Good catch, because you knew you were moving to RNA, and we use uracil instead of thymine. If you have the letter T in DNA, what do we move to? A. If we have a G? If we have a G? If C? Amazing. You guys are incredible. You guys know how to do transcription. You guys doing okay? Okay, then when it comes to translation, like I said, this is a bit more tricky. If you want to, those blue books that I gave you for study, they have a codon chart in the section for this. If you want to look over it, you certainly can. If you want to practice a few things, you certainly can. I can't do this from memory because I'm crazy, but go ahead.
Do they expect us to know that on the EOC?
Like memorize it? Yeah. No. Like they will give you a codon chart. You know what I mean? Like that would be insane. I doubt even some scientists that work with us all the time would even bother to memorize it because they just reference it. Though I'm sure if you do it enough, it probably sticks in there. But anyway, we're basically moving RNA to amino acids, right? We're building up the primary structure of a protein by doing this. So let's just say I've got letters like A, U, G, A, C, A, G. I don't know, I'm just making this up off the top of my head. Well, when we look there, AUG always codes for something called the start codon, right? Usually referred to as methionine. This is going to be the beginning of every single sequence that we ever copy. It says, hey, start copying here. That's the plan. That's the idea. Doing okay? Remember how you got to look on your chart, find the first letter, second letter, third letter. Doing all right? No questions? Okay. Let's go ahead and hit up them pedigrees. Pedigrees basically work like this. You have a square, you have a circle. Squares equal male, circles equal female. And you can halfway color this in, which is a carrier, and then you can all the way color this in, which means they have the trait. Now, of course, you could do this with sex-linked traits as well, but for now, let's just focus on the recessive traits. On your diagrams, I've drawn just blank ones, but let's go ahead and make a scenario for us. So I'm going to color in mom all the way, and I'm going to go ahead and color in dad all the way. Basically, what this tells me is I know their genotypes. If they have it all the way colored in, they're affected by this trait. And I'm just going to point out to you, this measures recessive traits, the presence of recessive traits. So that means this mom is little a, and this dad is also little a, little a. Do you guys understand so far? So when we go down and we branch these things off, which is a very ugly job on my part, the firstborn here is a female, circle, secondborn is a male, and the thirdborn is also a male. In this case, I know what letters can be sent from mom and dad, don't I? Mom can only send what kind of allele? Recessive. Dad can only send what kind of allele? So that means this child would also be all the way colored in. That'd actually be true of every single one of these kids. So if both parents are affected by this thing, they're all going to be affected by this thing. But let's go ahead and say this lady meets a man with normal whatever. I think I said previously like widow's peak. Widow's peak is like that point on your forehead for hair. So let's say all these parents and all these kids, all their children, had a widow's peak. But in this case, this guy, he doesn't have a widow's peak, and nobody in his family ever did. So this would mean he has big A, big A. When we look down at their kids, I believe it's female, female, male. Oh, no! No, I can't go back.
It's all gone.
It is all gone. It says female, female, male. And we said one parent up here was like this, one parent was like this, right? Well, we know this is big A, and we know this is little a, little a. So when we look here, what this is going to show us is no matter what, I know for a fact at least half of this has to be colored in. Because mom in this case was a full-blown widow's peaker, right? Whereas dad here, he can only send a dominant allele. So in this case, if they're all heterozygotes, well then that means they're all going to have normal hairlines with no widow's peak, even though they would all become carriers. So, and a lot of the times, you won't fill this in randomly like what we did. A lot of the times they'll give this to you, and you'll have to analyze, hey, who's a carrier in the third generation? So meaning grandparents were up here, parents are down here, grandkids are down here. That would be the third generation. And when they point out things, you can find out who they are. How you guys doing? Questions? Sorry for the little boo-boo there. Biotechnology. Just to point out to you, biotechnology is basically whenever we take one gene from an organism and transfer it to another organism, usually because we think that's cool and maybe there's a benefit behind it. Like we've done it with all kinds of foods nowadays. We've genetically modified them so that they maybe yield more stuff or give us more nutrition or do all kinds of crazy things. So let's go ahead and talk about this big project that happened, the Human Genome Project. I'm going to get lazy and write human. This was our sequencing of all of human DNA. So what does this mean? Well, it basically means we looked at all the possible genes inside of humans. We looked at a lot of people's genes and mapped out, hey, this seems to be where hair color is found. Hey, this seems to be where height is found. Hey, this seems to be where Alzheimer's is found. Stuff like that has helped us understand and research what can we do to change these things and in turn to change what the results could be in a person. So this is playing a huge role today because it helps like give us the basis of knowledge for what we need to modify. Then there's the restriction enzymes. Restriction enzymes. These restriction enzymes allow us to cut DNA. So when we want to cut and modify things, well, we have to do it precisely. Restriction enzymes are basically brought into there and basically helps us excise the section that we want. Usually in a very precise fashion. Not the most precise fashion is actually reserved for something else, but we'll talk about that here in a second. Then we have PCR, which stands for polymerase chain reactions. This helps us copy a lot of DNA in a lab. If you've ever seen like a forensics research program or anything like that, or even TV, they say they got to run DNA samples in a lab because what they're doing is they're making tons of copies of that stuff because you'd only be able to test it once if it was just one strand of DNA. So they try to extract that, run copies of it, and once they do that, well then dang, they're good to go. They can run as many tests as they need to because it's exact copies of the DNA that was there before. And then we have this cool thing called CRISPR. It's not misspelled here. There's no E at the end. It's CRISPR. Huh? Yeah, essentially. This allows us to modify DNA precisely while organisms live. Now, designer babies is more like, hey, before the baby's a baby, we're going to like tweak it and modify a few things. And we can't tweak everything. And it's still not like a thing you can legally do. So don't worry about that. But CRISPR is based... Yeah, you can't do designer baby things. Anyway, CRISPR is basically maybe the best version of gene therapy we can think of. The idea is basically we can release all these restriction enzymes and use this really precise cutting method to basically cut it and insert it into what we want while the organism lives, which means you don't have to do this beforehand, you can do it after the fact. So if somebody's born with a disease, CRISPR might be able to cut out the disease and put in a normal gene sequence, which means they would be cured essentially of that disease. That's a bit of an oversimplification here, but that's basically the concept. That's remarkable. Or even, what if it built in immunities for some diseases into us already? So we couldn't have to worry about like getting a vaccine. What if we just gave you one vaccine and it would take care of it on a genetic level instead of making your body go through another response? That's insane. That's crazy. Anyway, moving on. Flipping pages. Flipping pages. Okay, so mutations. Mutations are really coming in three varieties. We have chromosomes, or chromosome mutations. I'm just going to say chromosome. Chromosome mutations, these basically are massive changes in DNA. We're not saying like the sequence is different. We're saying like entire long threads, like entire chromosomes, big chunks, are either being destroyed or replicated or duplicated or flipped upside down, put in the wrong order. It influences a lot all at the same time. You've probably heard of something like Down syndrome, which leads to a lot of differences for that person, right? They might have physical differences, they'll probably have a lot of mental differences, and that's from having one extra chromosome. So that would be a chromosomal mutation. Frameshift mutations, while it does affect less DNA overall, it can still be quite devastating. It can have a major effect on protein production. So we have two versions. We have insertion and we have deletion. I think it's pretty obvious. This is when we add a nucleotide, and deletion is going to when we delete a nucleotide. Now, seeing the effects of that means when we're reading our codons, the three-letter combos of these things, we're shifting where they originally were, which makes it read something else. So let's say we have this letter combo, AUG, CCG, and I don't know, UAA, whatever. It doesn't really matter. Well, with insertion, let's say we add in a new letter here, and we add in a C. So now our sequence becomes AUC, GCC, and then GUAA. So instead of just reading AUG, now we read AUC, which probably won't code for the same thing. Instead of coding for CCG, we're coding for GCC, which is not coding for the same thing. So this would be a pretty devastating effect. With deletion, it's the same thing, but we're deleting a letter. So everything shifts over to the left to fill that gap, and of course things get read improperly again. How you guys doing? Okay. And then we've got point mutations. Point mutations are in three versions. Let's go ahead and say we're looking at AUG, right? Which makes a start codon. But let's say we've got a silent mutation, a missense mutation, and a nonsense mutation. A silent mutation happens and it doesn't change the overall protein. I should actually do a different example. I really don't know any off the top of my head, so I'll just make one up. Let's say AAA makes valine. Whatever. It might be true. So if we change the last letter here and say make it AAC, well, this will probably still make valine. So if the last letter is changed here, it's probably more safe. Yeah, there could be a change that's something different, but it's silent because it makes the exact same amino acid. So we'll just say same AA. When it's a missense mutation, what happens is usually it's a middle letter that's changed, or it could be the last letter. So in this case, we're going to make AUA. So if that middle letter changes, or in some cases the last letter, let's say it makes serine instead. Okay, well that's going to mean we make a different amino acid, which of course is going to make it a lot harder for us in terms of basically understanding kind of what's going on. Are we going to make the same protein? Is it going to be slightly different? What's going on? And nonsense is pretty impressive. So if it becomes UAA, and we change that front letter, in most cases this means we could make a stop codon. And if a stop happens, we're not making a protein. It stopped. It didn't finish the job, which of course would be devastating, because if no protein's formed, then we can't, you know, digest milk, or I don't know, you could have sickle cell anemia, like all kinds of things. I don't want to say that not being able to digest milk is devastating. It's okay. That's pretty light in terms of the genetic roll of the dice. Anyway, so moving on. Cool. Classification. Whew. Okay, so classification, we've got domain, kingdom, phylum, class, order, family, genus, and species. Just to point out to you, these 7 down here were actually created by a guy named Carl Linnaeus. I believe he's a Swede. He basically came up with this classification system we still use today. He also came up with naming organisms using these last 2 words that we refer to as binomial nomenclature. And binomial nomenclature is what we see as our names, right? So Homo sapiens, Canis lupus. Generally speaking, you capitalize the genus name, you lowercase the species name. Anyway, this also leads us to seeing something like domains. They come in only 3. We only have 3 domains currently, which he didn't know at the time because we've created that since then. But there's 2 that really matter to us. First off, there's eukarya, which we'll talk about here in a second, but bacteria and archaea, both of these are prokaryotes. And this is important to remember because they're really simplistic organisms. That's the main concept. When it comes to eukaryotes, eukaryotes come in 4 kingdoms. These 4 kingdoms, of course, are the animal kingdom, which is basically anything with a face. Plant kingdoms, which is basically anything that's has roots and grows in the ground. Excuse me. Fungi, which are a lot like plants, but they're probably not green and they have a different kind of texture in their cell wall. And then there's the protists, which are the weirdos. They freaky, and they do a lot of weird stuff, and they are hard to kind of pin down in terms of their definition. But if we're not exactly sure, we put them in that weird group. These 4 kingdoms, and not mention archaea has a kingdom, it's called archaea, and bacteria has a kingdom, it's called bacteria. Pretty easy to remember that one. They are different from each other in a lot of different ways, but this leads to that concept of taxonomy, which is basically classifying organisms into groups. And taxa is the phrase that we use to say, oh, that's a group. So there you go. Taxonomy, putting things into different groups. How you doing? Any questions? Are you sure? All right, groovy. Origins of life. This is rather short, which is fine with me. But Earth's age is, generally speaking, 4.6 billion years old. Life is probably about 3.6 billion years old, but there might be some evidence out there that says it might be closer to 4. So that's some pretty interesting stuff. I think there might even be fossils that might be dated to 3.8, but we're still trying to figure some stuff out. That's cool. That's really neat. Fun fact also, we don't think fossils can be older than 4 billion years old because they'll literally fall apart before that period of time. But anyway, that might change in 20 years when we find something really cool. Prokaryotes came before eukaryotes. This actually might bring up something called the Miller-Urey experiment. Do you guys remember this? We said there was like these scientists that basically put a whole bunch of non-organic things inside of a vacuum, and they added some electricity with some water. There was no oxygen, and from that they created amino acids. That idea of like a warm, nice place, a nice warm spot on Earth that was wet and allowed like electricity show up could give the spark to life and start forming amino acids. Amino acids, when they link together, form proteins, and proteins are self-replicating in a lot of cases. So if they self-replicate and keep doing that, it's basically trial and error until they start spitting out some other weird stuff. And carbohydrates probably existed independently as well, and dang, maybe even lipids. So now all of a sudden we're forming things together. Holy crap, we started forming the building blocks of life, which means maybe they started self-replicating as a whole and formed life. Now, I also want to point out photosynthesis came very early on. It was necessary because some of the first living things used photosynthesis to survive. Their only main energy source was the sun. They didn't have completed sugars to be able to process. They had to make them themselves. And then it says complex life had to form afterwards. We're talking about animals, you know, like more complex organisms, land animals, and then it just rapidly gets more complex as we move forward, which explains why 65 million years ago, not that long ago, we have a giant extinction event for a lot of the land dinosaurs, and yet here we are walking around, mammals have kind of taken over. That's pretty cool. So we've gotten more complex over time. But I say all this just to get to this idea of endosymbiotic theory. Endosymbiotic theory is basically prokaryotes absorb other prokaryotes to form first eukaryotes. This was really proposed back in the 1970s, and since then it makes a lot of sense because chloroplast and mitochondria in our cells, or modern eukaryotic cells, those components have their own DNA, have their own ability to produce energy. They are independent living things if they were found outside of the body. And cyanobacteria seem to be a lot like those. So they've probably evolved to live inside of a bigger prokaryote, and since they depended on each other, we saw the first basically the first cell organelles that were membrane bound show up, which also explains why mitochondrial DNA is unique as it gets passed along. As a matter of fact, it's maternal DNA. So endosymbiotic theory, pretty big concept. Any questions? No? Okay, here's the really scary one. It took me a long time to figure out what I was going to do for this space because it's natural selection, evolution is a really big concept. So let me walk you through this crazy complicated mess that's on your paper. So natural selection. There's not a ton to write here, but let's just make sure we can talk about these phrases and ideas. First off, evolution, I've already defined it for you. We said it's change over time in a population, and that can be big changes or small changes. It can happen quickly or slowly. Evolution can happen pretty quick. We usually call that microevolution, and big, slow, classic evolution is usually referred to as macroevolution. But the big concept of natural selection, sorry. The big concept of natural selection is basically that phrase that says benefit, okay? Or basically traits that become more common, and I think I wrote the word useful there. The most useful or beneficial traits become more common in a population. That's the main driving force behind evolution. That's what Darwin proposed for the first time ever, and people were like, yeah, maybe change is happening, but now we know why it's happening, because there are genes and traits that are found in things, and if it gives them an advantage to live in an area, then they're more likely to live, which means those traits will get passed on, because the ones with the bad traits probably didn't have as many offspring. if they even survived. So the better traits seem to win. So I kind of did this weird formula thing. So we've got limited resources, and we've got overproduction. Okay? These two things together lead to stuff like overpopulation. That means having a lot of babies. Remember that? We said organisms can sometimes make a ton of babies, like frogs. They make tons and tons and tons of baby frogs all at once because they know a lot of them won't survive. Sea turtles do the same thing. A lot of them won't survive because there's limited resources and there's just overproduction in terms of things. If we add this with genetic variation, which is caused by sexual reproduction and even mutations, I'll add that in there. Okay? And like reproduction. Remember how meiosis makes variety? So if we get genetic variation and we mix that together with these limited resources and stuff, that leads to competition. Okay, competition basically means organisms start competing with one another, even if they're the same species, or competing with other species, intraspecific instead of interspecific relations, and this leads to selection. Selection means one trait seems to be the better one. So that leads to survival of the fittest. And then this leads to what we call an adaptation. So these adaptations help them better survive their environment. And this is kind of the result. So all of these things added together, they kind of fit together with time, which means many generations. Okay? Multiple generations. Like we said, microevolution happens on a small scale, but that's because bacteria reproduce really, 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 really fast. So in 20 minutes, you can get a new generation for E. coli. So if you wait 2 months of letting them reproduce, you could see a massive change in a short period of time. So really what we're saying is generations. But as we do that, that leads to natural selection. All those things together is what causes natural selection. Now, there is a note at the bottom with a little star. Please take note of this. It says you've got things like gene flow and genetic drift. These two things really affect small populations, like endangered species, or you know, populations in like zoos or in captivity or in like nature preserves or conservation areas. Like we need to keep this stuff in mind because gene flow is migration. That's what that is. We're just saying populations mix together with other populations, and that might add in some variety in there. And genetic drift is when we suddenly kill off a lot of them. In big populations, genetic drift doesn't have a huge effect. But if you have a really small population, like the key deer down in the Florida Keys, when Hurricane Irma was kind of going through there, we were nervous because there's not many key deer. What if that hurricane would have killed off a lot of them, if not all of them? Well, then the population may never be able to recover. Still, there's a situation with them. That's why they're endangered. But you're catching on, right? You get the idea. This has an effect on small populations. How you guys doing? Last box of the day. So when we're talking about evolution, we really care about the evolution evidence. There's a ton more than what we're just talking about, but we've got embryology. This is when we study or compare embryos from one organism to another. Early development looks mostly the same if it's between animals. But when we start to get a few weeks past a certain point, sharks and humans don't look much like each other anymore. Lizards and humans don't look much like each other anymore. But apes and humans, they still look a lot like each other at the fetal stages. So when we can see that a similarity, that maybe shows closeness in terms of relatedness. There's also homology. And you guys will remember this is comparing body structures. So we have homologous structures and vestigial structures. And analogous structures. I just want you to remember those terms there. Vestigial are like leftovers from evolution. So we have a hip bone, so do whales, but they don't have legs. So it probably shows we had a common ancestor at some point where both of us had legs. Analogous structures mean they have the same function, but they don't have the same structure. So like butterfly wings and bird wings, which means they're probably not closely related. Homologous structures mean same structure, different function. So like a cat's paw and a human's hand. They use it for different things, but they basically have still the same bones in there, which shows we probably do have a common ancestor. DNA molecular evidence, I'm just going to put a star next to this one. This is the strongest form of evidence that supports this. Between humans and chimpanzees, there's hardly any difference in DNA at all. It's like 99.8% or 98%. It's really, really, really close. When it comes to humans and rabbits, there's a bigger gap. When it's humans and fish, it's a bigger gap. When it's humans and bacteria, it's a huge gap. But the thing is, if we can look at DNA sequencing and we can do that with other organisms, we can compare them to each other, and you guys did that in class as well. When it comes to fossils, this kind of does connect to homology because you're looking at bones, you're looking at parts that are left behind, and you're comparing those structures to each other. The cool thing is it gives us a timeline of change. And then, of course, we have biogeography. And biogeography is basically our ability to say like, hey, this organism was found growing in this area. Maybe these conditions influenced why they look the way they do. And then, of course, with continental drift, maybe like South America and Africa have the same bones on them, and as they split over time, we were able to see that the same bones were found on the different continents, which shows maybe we can see what conditions led to the creation of those species. Oh gosh. So do you guys have any questions there for the whole thing? Any questions on the whole sheet? You sure?
Absolutely.
Okay.